Welcome. Immune response to pathogen. Very important to understand the immune system and its component and how it deal with the pathogen, either bacteria or virus. The cells involved in the immune response. Overview: site occupied by the pathogen, where we can find the pathogen in the body. Antibody responses, cell-mediated responses, population of T cells types, specificity of the immune response, cell involved in the immune response, diversity of receptors, specificity and classes of the major histocompatibility, complex major histocompatibility molecules and its type 1 and type 2, cells of the immune cells and the origin of it, B cells and T cells, lymphocyte recirculation in the body, and leukocyte migration and localization when it finds any pathogen, how deal with it. Site occupied by the pathogen, extracellular. Site of the most bacteria, we find bacteria extracellular, and this is elicits antibody humoral response. Intracellular, this is the site for the virus and some bacteria. Elicit cell-mediated response. Types of antibody effectiveness: neutralization of antibody, neutralize toxin, bind to attachment molecule, opsonization, antibody bind to the pathogen, and surface blocking, complement activation occur on the antibody bound to the pathogen. Neutralization, here the antibody neutralize the toxin or bacterial toxin or the macrophage make forming phagosome, and this is neutralize the toxin, get inside the macrophage and lysis. Opsonization, the extracellular bacteria, and this is the macrophage. Ingestion by the macrophage, digestion by lysosome. The complement activation, bacteria in the plasma here, and this is activate the complement system. C1 appear, and it's proteins, types of proteins inside the blood, and it's activated by presence of the toxins or any bacteria, and the cascade complete continue. C2 appear, C4 appear, and this is complex occur digestion in the lysosome, lysis and ingestion. Common fate of the pathogen or toxin after neutralization, opsonization or complement activation. Pc or complement receptor on the phagocytic cell binding pathogen and toxin complex with antibody. Endocytosed complex fuse with the lysosome containing acid hydrolases, complex digested by the lysosomal hydrolases. Complex digested by lysosomal hydrolases. Fate of antibody toxin or antibody pathogen complex. Here we find pathogen fuse with the lysosome, and the antigen antibody complex is digested by lysosomal hydrolysis inside the lysosome, and this is the phagosome. Cell-mediated response to intracellular compartment, cytosolic or vesicular. Cytosol and the nucleus connected via nuclear hosts, site of the virus and some bacteria. Vesicular membrane bound in types in the plasmic reticulum, ribosomes, lysosomes, Golgi apparatus, site of some bacteria and some parasites. Location of the pathogen determine which T cells population response. Cytosolic T cells that express CD8. The CD8 is the protein outside the T cells. Vesicular subpopulation of helper T cells, T helper 1 that express CD4, extracellular subpopulation of the helper T cell, T helper 2 that express CD4. Cytotoxic T cells, here the virus infected cells, and the cell express viral antigen outside, and the cytotoxic T cells, infected cell is killed by the cytotoxic T cells by activation of the nucleases that cleavage host and viral DNA. Helper T1 cells, here microbacteria inside the macrophage, and here is the antigen presenting on the macrophage. Macrophages activated infected macrophages. T helper recognize the macrophages. Specificity of the immune response reside in the receptors. T receptors recognize peptide associated with major histocompatibility complex and is univalent. B cell receptor surface immunoglobulin recognize antigen and is bivalent. Here the B cell receptor, diversity of receptor specificity repertoire. Historically, two different hypotheses to explain diversity: instructional template and clonal selection. Structural hypothesis, although simpler, doesn't explain how host distinguish self from non-self antigen. Four basic principles of the clonal selection: each lymphocyte being a single type of receptor of a unique specificity. Interaction between a foreign molecule and the lymphocyte receptor capable of binding that molecule with high affinity lead to lymphocyte activation. Differential effector cells derived from an activated lymphocyte will be receptor of identical specificity to those of parental cell from which the lymphocyte was derived. Lymphocyte-bearing receptors for self-molecule are deleted at any early stage in lymphoid cell development. Class 1 major histocompatibility molecules expressed on the surface of all nucleated cells, recognized by T cell receptors of cytotoxic T cells. CD8 bind to class 1 major histocompatibility peptide complex. Source of peptide is cytosolic compartment. Classes 2 major histocompatibility molecules expressed on the surface of some nucleated cell, mainly antigen presenting cell. Recognized by the T cell receptor of helper T1 cells. CD4 bind to class 2 major histocompatibility peptide complex. Source of peptide is vesicular compartment. Cells expressing class 1 and class 2 major histocompatibility. All nucleated cells express class 1 major histocompatibility. Cells expressing class 2 major histocompatibility also express class 1 major histocompatibility, MHC. Non-specific and specific immune contrast. Non-specific, natural and native or innate system is placed prior to exposure to antigen, lacks discrimination among antigen, can be enhanced after exposure to antigen through effects of cytokines. Non-specific and specific immunity. Specific, acquired and adaptive immunity is induced and enhanced by antigen, shows fine discrimination and has memory. Cells of the immune system all derived from the bone marrow. Two main lineage derived from the bone marrow: hematopoietic stem cells, lymphoid lineage, the T cells, B cells, natural killer cells; myeloid lineage, monocytes, macrophages, dendritic cells, megakaryocytes, granulocytes. This is the hematobiosis. Here the myeloid progenitor. Here the monocytes, macrophage, mast cells and granulocytes, and here the megakaryocytes, which is the parent cells of the platelets. Here the lymphoid progenitors, the natural killer cells, the B cells, T cells, and the hematopoietic stem cells. Lymphocyte recirculation. Secondary lymphoid tissue, lymph node and spleen, main site where lymphocyte encounter antigen. frequent lymphocyte having a receptor specific for a given antigen is low. Recirculation of lymphocyte through lymphoid tissue optimizes productive encounter with antigen to initiate response. Lymphocyte recirculation is the system of the lymph system. Naive lymphocyte enter lymph nodes from the blood circulation. Lymphocyte return to blood via the thoracic duct, and the antigen from the infected area go to the lymph node via the lymphatic system. Leukocyte migration and localization. Bone marrow and thymus, the primary lymphoid tissue, produce B cells and T cells respectively. The thymus produce T cells and the bone marrow produce the B cells. B cells and T cells recirculate through the spleen and lymph node. Antigen presenting cells pick up antigen and migrate to secondary lymphoid tissue and interact with the T cells and B cells. Leukocyte migration and localization on the bone marrow. The thymus is the source and the drainer of T cells, and the bone marrow the source of B cells, and the secondary lymphoid organs, the spleen and the lymph node, and here the antigen presenting cell, and this is the dendritic cell, the macrophage, as is the scale of the circulation of the T cells and B cells.